Hi everyone, thank you for your interest in the poster titled Quantitative Bioanalysis for HT-KIT Vivo-Morpholino using hybridization LC-MS presented at the 2024 OTS conference in Montreal. My name is Mingluan Chen. I am the principal research scientist at Altasciences, and I will be my pleasure to discuss our research with you today. Vivo-Morpholinos are a type of conjugated PMO. The structure is highlighted in Figure 1. This molecule can enhance cellular uptake and stability, making them essential for gene function studies, disease modeling, and treatment development. To resolve issues with traditional chemical extractions and chronographic resolution, and in this study, we developed a local hybridization LC-MS method to quantify HT-KIT, a novel Vivo-Morpholino for cancer treatment. As shown in Figure 2, the workflow involved digesting mouse serum samples and hybridizing them with the bioactinated capture probe, and the analytes probe beads complex was formed, washed, and thermally denatured to release HT-KIT. LC-MS analysis was performed using a SciX 6500 plus triple quadruple MS in positive ESI MRM mode. To optimize the mass performance, we first adjust peak asymmetry of positively charged HT-KIT on negatively charged signal LC colons by using orders X select CSH C18 colon and the ending HFBA in the mobile phase. Ammonium bicarbonate was used as a post-colon addition to enhance the MS ionization. As a result, the baseline separation of HT-KIT and the internal standard was achieved with the LLQ at 1 nanogram per ml using 25 microliter mouse serum samples. In hybridization extraction, the best recovery was obtained with a full-length capture probe and the sativa beads, which reduced long-species binding of the HT-KIT on beads. The calibration curve ranging from 1 to 250 nanogram per ml was linear based on the peak area ratio of HT-KIT to the linear PMO internal standard. Representative chromatograms are shown in Figure 3. Precision and accuracy were verified within 4 QC levels, meeting accepted criteria listed in Table 1. The assay was specific and free of matrix effect, as highlighted in Table 2. In summary, the developed hybridization LC-MS method is both sensitive and selective, meeting all bioanalytical criteria for quantifying HT-KIT in mouse serum. This advancement will significantly contribute to the research into PMO therapeutics. Thank you very much for your attention, and please do not hesitate to reach out if you have any questions.